So this is the technique that I use for patching the cells to do the transformation lab. Once they've grown on a petri dish as single colonies, I take a sterile toothpick and pick up a colony and move it to a new LB plate where I scrape the cells in a small square. So I scrape them left to right and turn the plate 90 degrees and scrape them left to right. And they will grow as a small patch like that is big enough, generating enough cells that the students then can use in the transformation lab. After an overnight growth, each of these patches will look something like that. And you can grow these 6 on a plate or so. I would keep the strains 4-1 and 4-2 on separate plates, partially just to keep things from getting cross-contaminated, and also just a little bit easier for the students to find their cells that they need when they run their labs.